Welcome to the lecture on Chapter 5, Day 2. Before we get into the new material for today, let's just do a quick review of what we've seen in the first part, in Day 1 of Chapter 5. So the first thing we talked about was electrical versus chemical synapses. And so we went into the structure of an electrical synapse. We looked at how electrical synapses are formed by gap junctions, which are made up of proteins, and that these proteins come together to form the gap junctions that allow ions to pass directly from one cell to the other. And so electricity or current can flow from one cell to the other directly. That's an electrical synapse. The next thing we learned about was chemical synapses, and we spent a lot more time on those and dived a lot more deeply into how those work. We learned that in chemical synapses, the presynaptic neuron contains vesicles that are filled with neurotransmitter, and that when that presynaptic neuron is stimulated by an action potential, it releases neurotransmitter into the synaptic cleft, which is a space between the presynaptic and postsynaptic neuron. That neurotransmitter then diffuses across the cleft to the postsynaptic neuron where it binds to receptors and activates them. We also learned about the different kinds of neurotransmitters that we can find in vesicles. We talked about amino acid neurotransmitters, GABA, glutamate, and glycine, and we talked about amine neurotransmitters, dopamine, serotonin, epinephrine, norepinephrine, acetylcholine. And we also talked about peptide neurotransmitters, which are found in the dense core granules or dense core vesicles in the presynaptic terminal. We also learned about the different configurations that a chemical synapse can have. In a chemical synapse, you can have axon synapsing onto dendrites or onto the soma or even onto other axons. We learned about the neuromuscular junction and how this is a special kind of synapse that has taught us a lot about how chemical synapses work in general, and has taught us how the neurotransmitters are released from the synapse. And we learned that the big chemical or the big ion that's involved in neurotransmitter release is calcium. And we learned how when there is an action potential that travels down into the axon terminal, the axon terminal depolarizes, and that opens up voltage-gated calcium channels and allows calcium in, which then causes the fusion of vesicles to the presynaptic membrane there at the active zone and allows the neurotransmitter to be exocytosed into the synaptic cleft. We also learned how the different types of neurotransmitters, the peptides, the amino acids, and the amines, are produced and where they're produced in the cell and how they are transported to the axon terminal in the case of peptide neurotransmitters, or how they are loaded into the vesicles in the case of amines and amino acid neurotransmitter. Now, in this part of the chapter, we're going to make it across the cleft and start talking about what happens in the postsynaptic neuron. So this again is the presynaptic neuron, the postsynaptic neuron, and we're going to start learning about what happens when neurotransmitters, which are released from the vesicles from the presynaptic neuron, bind to and activate receptors in the postsynaptic membrane. So we're going to talk about different types of receptors and the effects they can have when they're activated. Then, to finish up this lecture, we will talk about synaptic integration, which is the beginnings of a discussion about how a postsynaptic neuron can compute the information, the input it's getting from a presynaptic neuron. And so we'll learn about how the postsynaptic neuron can react differently depending on the types of signals that the presynaptic neuron sends out, and how this postsynaptic neuron actually makes a computation because it receives multiple inputs from multiple axons, and so it needs to integrate all of that information to decide whether to have an output in the form of an action potential. But let's go back to what the first part of our discussion here and talk about the types of receptors that you will find on the postsynaptic membrane that are activated by neurotransmitters. The two big families of receptor types that we'll find on the postsynaptic membrane are ionotropic and metabotropic receptors. That's an M, metabotropic. Ionotropic receptors are in your book called ligand-gated ion channels. And in your book, metabotropic receptors are also called G-protein coupled receptors. Let's talk about receptors and ligands for a minute. So a receptor, whether it be ionotropic or metabotropic, is a membrane protein or a complex of membrane proteins that come together to form a receptor. So it can either be a single polypeptide chain, that is a receptor, or it can be multiple polypeptide chains that come together as subunits to form a receptor. But when we talk about receptors, they have to receive something, right? And the thing that they receive is called a ligand. So a ligand is the chemical that binds to receptors to activate them. You can think of a receptor kind of like a lock, and a ligand would be like the key. And so when the key goes into the receptor, it changes the shape of the receptor. And when it does that, it causes the receptor to activate. Now we're going to see that that activation can take a couple of different forms. In one form, its activation can open a door. In another case, though, the turning the key in the lock won't open a door, but it will activate some mechanism inside the cell. And so, but in both cases, with a receptor, that's the lock, and the ligand is the key. You've maybe heard of the word ligature, which is to tie things together. And so a ligand, that word comes from the idea that this chemical binds to or attaches to the receptor. In the case of chemical synapses, the ligand is the neurotransmitter. And so this ligand will be released from the vesicles in the presynaptic neuron and travel across the synaptic cleft and bind to its receptor on the postsynaptic membrane. Another very important thing to recognize is that receptors typically only bind a single kind of ligand. So if you think about a lock, only one type of key can fit into that lock. My car keys won't fit into my door lock. And so receptors are very specific for the ligand that they bind. But there's a flip side to this, which is that the ligands can actually bind to multiple kinds of receptors. So ligands can bind to multiple receptors, to multiple kinds of receptors. So with the key lock analogy, although my lock can only take one type of key, that key that I have can open many doors. So you may have a key that can open your front door, or it can open your back door. Or you may have a key that turns the ignition on in your car, but it also can open your trunk. So in the case of something, so take your car key, it can both, that ligand, that key, can both activate a mechanism by turning on your engine. It can also open a door by opening your trunk or opening your passenger side door. So that key has multiple locks that it can open. But those locks, each one of those locks can only take one kind of key. They can only be activated by one kind of key. So the receptors are specific for the ligand that binds to them, but ligands can bind to multiple types of receptors. Okay, I got to this figure and realized that the name I had just given you about ionotropic receptor was not exactly what the book said. The book actually calls it a transmitter-gated ion channel. But this is the same thing as a ligand-gated, and it's the same thing as an ionotropic. So let's take a minute and talk about these. These type of receptors are the doors in my analogy. So when a neurotransmitter binds to these receptors, a channel is opened. So as you can see, these guys are gated. This is similar to what we saw with the voltage-gated ion channels that we learned about in Chapter 4. But now, instead of opening due to a depolarization in the membrane potential, now what we're going to see is that these open when they are bound by a ligand or a neurotransmitter, thus the name transmitter-gated. So the key here that opens them is the ligand, the neurotransmitter released from the presynaptic terminal. And they're gated, meaning that they will be closed unless the ligand is bound to them, just like the lock on your trunk or on your door. It's only opened when that key goes in and turns. Many of these channels are formed from multiple subunits. So in this case, the picture of this channel here is formed by 5 different subunits that come together, and there is a pore in the middle of them. And these are membrane proteins, and so they're going to traverse the membrane and allow ions to travel through them. This is where the word ionotropic comes from. Tropic just means that they allow change, and so in the case of ionotropic, the change we see is in ion flow. So what they're going to do is let ions travel through them when they open, and not when they're closed. And so these will be permeable to ions. Okay, there's some important ionotropic channels that you're going to be learning about, and I'm going to introduce them briefly to you now so that you can see them. But before I do that, I want to explain a concept to you called EPSP and IPSP. Okay, here we have a picture of a presynaptic neuron and a postsynaptic membrane, and we have let's say this is an ionotropic receptor. It's going to be it's a gated channel that will open when a neurotransmitter binds. So let's release neurotransmitter from the presynaptic neuron, and let's have it bind to the receptor. What do we mean when we say it binds? So there is a pocket on the receptor where the ligand or the neurotransmitter fits perfectly. And when the neurotransmitter fits into that pocket, the protein changes shape. The receptor does. So this ionotropic receptor changes shape. We call that a conformational change. And when that conformational change happens, the pore opens. Now there are different types of ionotropic receptors. Some are permeable to cations, and some are permeable to anions. And as you can imagine, if you're permeable to cations, you're going to let sodium and potassium through. And if you're permeable to anions, you're going to let chloride through. Now you might be asking, what about calcium? There are calcium channels too, but the main ones that we'll focus on in this class are sodium-potassium channels and chloride channels. And one of the first things that you're going to notice about these channels is that they are a little less selective than we saw for the voltage-gated channels and for the leak channels. So there are channels that are cation channels. Notice I didn't say they're sodium channels. I said they're cation channels. This means that they let sodium and potassium through. And so if I open that, sodium could come in to the cell through that channel, or potassium could leave. Those anion channels, typically, the only anion we really talk about is chloride. So when I say an anion channel, you can almost guarantee that it will be a chloride channel, but it'll let chloride through. So really, the main point here is that the selectivity of the cation channels is not as stringent as we saw with the voltage-gated channels. These guys will let sodium and potassium through. Okay, now let's think about what would happen if I opened up a cation channel. So a ligand binds, and let's say this is a cation channel. So the big types of cation channels that we're going to look at are called the acetylcholine receptors and the glutamate receptors. Now these two chemicals that I just named, you hopefully recognize those as neurotransmitters. And so these two neurotransmitters, acetylcholine and glutamate, they open cation channels. Now remember, they are keys. They can fit in multiple types of locks. So these aren't the only types of locks they can open. But for now, I'm talking about the ionotropic receptors that they bind to. Let me just briefly mention the anion channels. So the anion channels are gated by an amino acid neurotransmitter called GABA. So GABA receptors and glycine, which is another amino acid, glycine receptors. These, when they are ionotropic, when they're the ionotropic versions of GABA and glycine receptors are anion channels, and they will then let chloride through. But again, GABA and glycine can bind to other types of receptors too. We'll talk about those. So those will be the metabotropic receptors. Just like acetylcholine and glutamate can also bind to metabotropic receptors. But when we're talking about ionotropic receptors, acetylcholine and glutamate open cation channels, whereas GABA and glycine open anion channels. That's the main rule. You know, I should mention before we go any further, calcium is another cation, and we will learn about one channel that's actually very important that allows calcium through. So when you hear cation channel, we're remembering, oh, those aren't as specific. They can let multiple types of cations through. So let's look at what would happen if we open a cation channel. Okay, let's talk about this receptor, and let's call it an acetylcholine receptor. And so it's ionotropic, and so I know that it's going to let in cations. So what would happen if I had acetylcholine here, this neurotransmitter ligand, that bound to and activated that channel? What would it do? It would open. It's gated by acetylcholine. What would happen when it opens? So it's a cation channel. It's a cation channel, so it's going to let cations through. Now what do we have in terms of cations? Well, we have sodium, and sodium is higher outside and so has a driving force pushing it in. And we have potassium, which is inside and has a driving force pushing it out. And we also have calcium, which is high outside and has a driving force pushing it in. Now, it turns out that the only two ions that can go through the acetylcholine receptor are sodium and potassium. So we're going to leave out calcium for now. We'll learn about another receptor, like I just mentioned, later that will let calcium through. But this receptor happens to only let through sodium and potassium. It's actually called the nicotinic receptor. And it's found in your muscle cells. So this is actually the receptor in your muscles that the motor neuron releases neurotransmitter onto. So remember when we saw that picture of the motor end plate with those invaginations of the membrane, and we had all those receptors in there? Those were nicotinic acetylcholine receptors. You'll learn that term later, I think, in chapter 6, nicotinic. But let's think about what would happen if I opened up this channel then. So let me draw it in an open state here. Well, sodium could come in, and if it did, it would depolarize the cell, but potassium could also leave, and in that case, it would hyperpolarize the cell. So, what will happen if we open that channel? Well, the truth is, both will happen, but one will happen more than the other. Sodium will come in more than potassium will leave. Can you think of why that is? It has to do with the ionic driving force. So if the inside of the cell at rest is negative 70 millivolts, who feels a greater ionic driving force, sodium or potassium? Well, the ionic driving force is equal to the membrane potential minus the equilibrium potential for the ion. So in the case of sodium, its equilibrium potential is around 60, and the membrane potential is around negative 70. So 60 minus negative 70 is 130 for sodium. Sodium's ionic driving force. For potassium, though, it's much less. Wait, I switched these around. Sorry. This is the membrane potential. Negative 70 was the membrane potential, and 60 was the Nernst potential. So let me go back. It would actually be negative 70 minus 60, which is a negative 130. That's a big ionic driving force. If we look at potassium's ionic driving force, so for potassium, it would be equal to negative 70 minus negative 80. So negative 70 minus negative 80 is 10. So in terms of magnitude, this one is way bigger. This ionic driving force for sodium is way bigger. This is sodium's ionic driving force, and it's way bigger than potassium. So when we open that channel, sodium's kind of bully its way into the cell, and because it's going to be, there's such a powerful force driving sodium into the cell, potassium, even though there is a force pushing it out, it's not going to be able to really get out. So the overall effect of opening this channel is going to be to depolarize the cell, because sodium will come in way more than potassium can leave. So if we look at the membrane potential, we will see that the membrane potential depolarizes. Okay, so there's resting membrane potential, and then it will depolarize. This is called an EPSP, which stands for excitatory postsynaptic potential. So it's a potential, it's a change in membrane potential that's happening in the postsynaptic membrane, and it's excitatory because it's getting us closer to, it's depolarizing the neuron, which is something that could lead to an action potential. Now this postsynaptic membrane may be in the dendrite, so this EPSP may not cause an action potential, because to get an action potential, we have to get depolarization to threshold in the spike initiation zone way over in the axon hillock. But it is a little depolarization blip in a dendrite, and if it is combined with other EPSPs from other parts of the cell, that positive charge of the sodium diffusing in may be enough to depolarize the membrane at the axon hillock to threshold, and then the neuron would fire an action potential. But it's an EPSP. And so the EPSP will rise, we'll get a more positive membrane potential, but then we'll see it fall back down to rest. And the reason that would happen is because the channel was closed. So if the channel closes, then we'll return back to rest because that sodium won't be coming in anymore. What would cause the channel to close? The channel will close when the neurotransmitter releases from the receptor. So the neurotransmitter is like a key in a lock, and you know that when you put a key into a lock, the lock doesn't swallow up the key and you lose the key. You pull the key back out. When you do, though, the engine turns off or the door closes again. So in this case, what happens is that the neurotransmitter can bind, open the door, but then it can actually unbind, and then the door will close right back up. And so that's what we see with an EPSP, is we see a depolarization and then a repolarization because the key has been removed and the channel closes again. What if we instead of opening a cation channel, we open an anion channel with, I don't know, let's say GABA, which is a neurotransmitter that opens anion channels. What will happen there? Well, it will be permeable to chloride, and chloride is higher typically outside of the cell, and so it has a driving force pushing it into the cell. And so what chloride will typically do is go into the cell. Chloride's Nernst potential is around -40, I think. And so if the membrane potential is -70, then -70 minus 40 is equal to -30. And so there is a driving force. It's not huge, but there is a driving force of chloride to go into the cell. So this would be the ionic driving force of chloride. And so chloride will come in and it'll bring a negative charge in. In that case, the membrane potential will hyperpolarize. Okay? And then, of course, when the channel closes, it will repolarize back to resting membrane potential. But that hyperpolarization is called an IPSP, or inhibitory postsynaptic potential, because what it does is it gets us further away from threshold. If I have EPSPs in other dendrites, and I have an IPSP in this dendrite, then that chloride coming in can neutralize those other EPSPs and prevent that positive charge from reaching the axon hillock. And so IPSPs counteract EPSPs. What we're going to see is that the neuron will add up all of the EPSPs and IPSPs that it's getting on different parts of its dendrite and soma, and depending on how much of each they have, you may or may not depolarize the axon hillock to threshold. All right, this is a picture from your book showing that exact same thing that we just described, which was the EPSP. So you open up that neurotransmitter gated channel and you let in sodium. That will depolarize the membrane. That's an EPSP. Or you can open up the channel and let in chloride, which would hyperpolarize the membrane, leading to an IPSP, inhibitory postsynaptic potential. An EPSP, just to be clear, is not an action potential. This is just a depolarization that happens somewhere in the cell due to neurotransmitter binding at that part of the cell. It does not necessarily mean that we're going to get an action potential. It's not the same thing as an action potential. It's very similar to what we discussed in class. It's like a generator potential. You can think of the EPSP as a generator potential. In fact, that's what it is. It's a generator potential. So it's the same thing. Okay, we've finished our discussion for now about ionotropic receptors. Now we're ready to talk about metabotropic receptors. Again, another name for them is G-protein coupled receptors. So let's go back to the key analogy. So if an ionotropic receptor is like a door, like your car door, and the ligand is the key, then putting that ligand in the receptor opens a door. Metabotropic receptors are different. They still are a lock with a key, but in this case, the ligand will bind to the lock and go inside the lock. And when you turn it, though, a door doesn't open. There's no door opening here. Instead, what's happening is we'll see that will activate something inside of the cell. So it's kind of like turning the engine on. We're going to turn something on inside of the cell. No doors are opening. We will learn about metabotropic receptors in much greater detail in Chapter 6. So right now I'm just going to show you a picture from your book that describes them, gives you an overview of them. Okay, here's a picture of two different types of metabotropic receptors. So this is the metabotropic receptor right here, the green thing here. So it's called a metabotropic receptor, or aka a G protein coupled receptor, or GPCR. That's very common for you to see GPCR, so don't be surprised if I call them GPCRs in class. What are these things? They are also membrane proteins. As you can see, it's sitting in the membrane here, and it's a protein, and it's typically a single polypeptide chain, as opposed to the multiple subunits we saw with the ionotropic receptors. And there are many, many different kinds of these, and each one of them is responsive to one kind of neurotransmitter. Now, the neurotransmitters we've already looked at, the amines, the amino acids, and the peptides, can all bind to these types of receptors. Maybe I'll just take a moment to mention that, remember how we said that each lock can only have one key fit into it, but keys can open multiple locks. So the amino acid neurotransmitters, GABA, glutamate, glycine, can bind to both the ionotropic and the metabotropic receptors. The amines generally bind only to the metabotropic, but in the case of acetylcholine, they can also bind to ionotropic receptors. Acetylcholine binds to an ionotropic receptor we just learned about called the nicotinic receptor. The peptides, the neuropeptides, only bind to metabotropic receptors. So amino acids can bind to both kinds. Amines can bind to both kinds if you include acetylcholine, but most of them bind to metabotropic receptors, and peptides only bind to metabotropic receptors. So what happens when a neurotransmitter binds to a metabotropic receptor? We see the same thing happen that we saw with the ionotropic receptors, in that there is a conformational change in the protein. The protein changes shape when that key goes into it. The neurotransmitter binds, it changes shape. That shape change is going to lead to activation of another protein in the membrane that's on the inner side of the membrane, and it's called a G protein. And this G protein binds to a chemical called GTP, and that's where it gets the name G protein. But this G protein here is what we call a signaling protein. And it's going to change the cell by signaling to things and turning them on or off. So it's kind of like it's kind of dormant. Well, it's dormant when there's no neurotransmitter, but when the neurotransmitter binds to the GPCR, the GPCR then activates the G protein, and the G protein wakes up and starts changing things. It goes around and it binds to proteins and turns them on or off. So some of the proteins that it can bind to are actually other channels. And these are not neurotransmitter gated channels. These are now G protein gated channels. What this means is that they open or close in response to a G protein binding to them. That's how they open or close. So it's a third type of channel we've seen that's gated but now in an even different way than we've seen before. These G proteins might also bind to enzymes. Now, enzymes are proteins that they turn some substrate, chemical molecule, into another type of molecule. So they turn a substrate into a product. So enzymes turn substrate, which is just a molecule, into some other type of molecule. So maybe an enzyme would take ATP, the molecule ATP, and convert it to something called cyclic AMP. And an enzyme would do that conversion, or at least facilitate that conversion. And the G protein might turn that enzyme on. And so if the G protein, if the neurotransmitter binds to the GPCR and the GPCR activates the G protein, then the G protein would activate the enzyme and you would get more cyclic AMP. These enzymes that are activated by G proteins often are enzymes that produce something called second messengers. Second messengers are small molecules that can be produced in large amounts quickly and can signal to other places in the cell. So they're these chemicals that can be made in large amounts very quickly, and that serves as a signal to the cell. Oh, we are making lots of cyclic AMP. The cell is changing. Something is happening, right? And the cyclic AMP that gets made can go around and affect other things in the cell. So there are a number of different second messengers that can be produced. We'll learn about these in Chapter 6, but just to get you exposed to them right off the bat, one of them is called cyclic AMP, which we just talked about. Another one is called IP3. Another one is called diacylglycerol. Another one is calcium, actually calcium. When these are made in the cell or released into the cell, they can change the cell in lots of different ways. We're not talking about just changing the membrane potential. We're talking about changing the way the cell looks, the way it behaves. We're talking about things that the cell changes that can be long-term changes in the cell. When I open an ion channel and ions flow through, the membrane potential of the cell temporarily changes. But then when I close that channel, the membrane potential goes back to what it originally was. But metabotropic receptors are different. Their effects can be longer lasting. Just like when you turn that car on, you open and shut the car door, your car's not much different. But if you turn on the engine and then let it run and then turn it off, your car is different. It's warmer. The oil inside is less viscous. It's more fluid. There's some changes that happened. That's what a metabotropic receptor does. It changes the metabolism of the cell. It changes the way the cell is. Those changes are often longer term. That's where the name metabotropic comes from. It changes the metabolism, or the way the cell uses and deals with energy. It changes that. And so when these metabotropic receptors bind, they have longer lasting changes. And because the process of their activation requires activating the G protein and then the G protein activating other things, it's sometimes a somewhat slower process than simply opening a channel, letting ions through, and then closing the channel back up again. Another thing that I'll briefly mention is autoreceptors. This is in your book, and if we have a presynaptic neuron here, we're talking mostly about receptors on the postsynaptic neuron, but believe it or not, there are actually receptors that can be found on the presynaptic neuron too. These would be called autoreceptors. So the neuron here releases neurotransmitter, and most of it binds over here on the postsynaptic membrane, but some of it can diffuse over here and bind and activate those receptors too. And those receptors can then cause changes in the presynaptic neuron. In many cases, these are metabotropic receptors. And like your book said, the consequences or the effect of activating those receptors can vary. But one thought is that what they kind of do is they kind of inhibit the presynaptic neuron. So when this gets turned on, it kind of inhibits this neuron to release less neurotransmitter with the next action potential. And the reason for that might be, well, if this autoreceptor is getting activated, it might mean that there's too much neurotransmitter in the cleft. And if there's too much, then what we need to do is quiet down that presynaptic neuron so it doesn't release so much neurotransmitter. So an autoreceptor could be a mechanism by which the cell does that, quiets down that presynaptic neuron. Okay, so we've released neurotransmitter, and we've talked a lot about the receptors that neurotransmitter can bind to, but there's something very important here. When you want to do in our nervous system, when we send out signals, we want those signals to be received clearly, but then we want when we want to stop the signal, so if I want to activate a muscle briefly because I want to lift a weight, well, I don't need my muscle to continue to contract for years afterwards. I need to be able to relax my muscle, right? And so we need to be able to quickly turn on receptors, but we also need to be able to quickly inactivate them. And like I said, the way that you inactivate these receptors, both the ionotropic and metabotropic, is to have the neurotransmitter release from the receptor. Now, when it releases, it could just as easily bind back on and activate the receptor again. But we don't want that. We don't want that to happen. We need to get rid of the neurotransmitter. So there are various ways that the neurotransmitter can be removed from the synaptic cleft. The first way that it can be removed is it can simply just diffuse away. And so this is space, and they can just diffuse out of the synaptic cleft, and then they won't activate receptors anymore. All neurotransmitters will do this to some degree, because you just can't prevent them from diffusing away. That's just the natural thing they'll do. They're highly concentrated in the cleft, and so they will diffuse away to areas where they're where they're at a lower concentration. Now, for some neurotransmitters, that's the only way we can get rid of them, and that is true for the peptides. So for the neuropeptides, or the peptides, the only way we can get rid of them, get them out of the cleft, is for them to diffuse. Now, diffusion is fairly slow, and peptides are fairly big, and so that process happens fairly slowly for neuropeptides, and because of that, the effect of neuropeptides can last longer, because instead of diffusing away quickly, they can hang around longer and activate those receptors more. But all neurotransmitters can diffuse away to some degree. But for the other types of neurotransmitters, we have ways of getting rid of them even faster. And one of the other ways that you can do that is called reuptake. Now, this can either be done by the neuron itself or by astrocytes. But what we're doing in reuptake is we're actively transporting the neurotransmitter back into a cell so that it's out of the cleft. And so in reuptake, they are taken back into the axon terminal. These are going to be transport proteins then that will transport those neurotransmitters back into the cleft, back into the axon terminal or into the astrocyte. The way these guys work is that they are what we call symports, and they're like a revolving door. Remember how we talked about those revolving doors where when we talked about loading the vesicles, remember we talked about how hydrogen on the inside of the vesicle leaves the vesicle, and as it does, it pushes this transporter that pushes in neurotransmitter into the vesicles so that we can load the vesicles with neurotransmitter. We've got a similar thing happening here, except that the ions aren't moving in opposite directions, they're moving in the same direction. So in many cases what's happening is that sodium, which wants to come into the cell, does through these transporters, and in the process it helps push the neurotransmitter back into the cell. And then once in the cell, it can either be recycled and reused, or it can be degraded by enzymes that break it down. And that's how you can get rid of neurotransmitter. A third way that you can get rid of neurotransmitter is called enzymatic cleavage. And what's happening there is that there is some sort of enzyme in the synaptic cleft that can Pac-Man and chop up those neurotransmitters. So they're not actually diffusing away, they're getting broken down right there in the cleft. The main one, actually the only one that we'll talk about that does this is an enzyme called acetylcholinesterase, or ACHE. And this is an enzyme that, as you can probably guess, breaks down the neurotransmitter acetylcholine into acetic acid and choline. And once it breaks it down, it won't activate its receptors anymore. Now, interestingly, there are some nasty drugs, nasty chemicals called organophosphates, or you may have heard the term nerve agents, that can block the activity of acetylcholinesterase. What that does is it brings him off a line, and acetylcholine then will overactivate its receptors. And that's actually a problem, because if you don't remove the acetylcholine from the cleft, then those receptors can be activated so much that they actually get an error and turn off completely. And so when acetylcholinesterase is gone, you have too much acetylcholine in the synaptic cleft, and these receptors, these nicotinic receptors, become desensitized. And when that happens, that's bad, because then they can't work at all, even if you got rid of the acetylcholine. And so these nerve agents work by paralyzing you, because these receptors, there's too much acetylcholine, they get desensitized, and then they don't work anymore, and so you can't work your muscles anymore. And one of the most important muscles that you need to be able to work to stay alive is your diaphragm. And if that's paralyzed, then you won't be able to breathe and you'll suffocate. By the way, pesticides are often organophosphates and often work by blocking acetylcholinesterase from working. And that's how they kill the bugs. Okay, the next topic is neuropharmacology. And we will revisit this again and again throughout the semester, and especially in Chapter 6. But we're introducing you to the topic here in Chapter 5. Neuropharmacology is the study of how different drugs affect the nervous system. And this is very important when we talk about treating disease. What we'll find is a large chunk of mental diseases and mental disorders result from defects in chemical synapse transmission. They either result in defects in receptors or in the neurotransmitters, or in the downstream effects of the receptors being activated. And so many of the drugs that work to cure or treat these diseases will involve affecting neurotransmitter receptors. And so there are different types of drug. There are lots of different drugs that can affect receptors, but there are two main categories of drugs. And these are the antagonists and the agonists. An antagonist is a chemical that can block the effect of a receptor. And so what these typically do is they bind to the receptor, and they're like it's like putting a block inside the keyhole, and so the key can't fit in. Or it might be like It might be like putting a child-proof doorknob on the outside of the door. So you could put the key in, but you can't actually turn the knob. So they work by different mechanisms, but ultimately they result in the receptor not being able to be activated as much. Now, these antagonists can bind to the receptors to different degrees with different degrees of affinity, meaning that some antagonists can bind to the receptors so tightly that it's basically a permanent inactivation of that receptor. So that's why that's why toxins from venomous animals can be so deadly, is because they are antagonists and they bind so tightly to the receptor that they don't release, and so that receptor is basically offline. And so if you have enough of that venom in you, many of your receptors could be completely taken offline, and then they won't work. So then, you're in big trouble. So antagonists are inhibitors. An example is curare, which inhibits the acetylcholine receptor, the nicotinic acetylcholine receptor. And agonists then are drugs that mimic the actions of the natural neurotransmitter. So they bind to the receptor and actually activate it. And they're not the neurotransmitter. They're not found in your body, but they're a drug that your body could take and they could activate it. So nicotine is an example of an agonist. It actually is an agonist of the nicotinic acetylcholine receptor. And so when you smoke and you get nicotine into your blood, it activates nicotinic acetylcholine receptors, even though you don't normally have nicotine in your body unless you smoke. But it's not a naturally occurring substance in your body like acetylcholine is. Acetylcholine is the natural ligand. Nicotine is the agonist, drug agonist that you could activate those receptors with also. Now, you might think, okay, so an agonist is good because it activates, and an antagonist is bad because it inactivates. And so agonists would be used for drugs to help people because it activates. Well, that's not necessarily true, because some diseases, like for example schizophrenia, are thought to result from too much activation of receptors, like dopamine receptors. So if you have too much activation of a receptor, then to get back to normal you need an antagonist. So it depends on how the disease works, whether an antagonist or an agonist would work best to treat that disorder. The next topic in Chapter 5 is synaptic integration. So up to this point in the chapter, we have been learning about what happens at a single synapse. We've talked about what happens in the presynaptic terminal as neurotransmitters get packaged into vesicles and they're released into the cleft. And we've talked about the receptors on the other side, on the postsynaptic membrane. We've talked about what kind of receptors are found there, and we've learned a little bit about how what they do when they're activated. Now I want us to take a zoom out and look at the whole neuron and think about the fact that a neuron is receiving input from hundreds, maybe even thousands of different synapses. So if I draw a neuron here with its axon, it's kind of a crummy looking neuron, but it's got multiple dendrites here. And these dendrites, there may be hundreds to even thousands of synapses on these dendrites. I don't have time to draw nearly that many, but these synapses could be, these axons that are synapsing on these dendrites could be coming from many different types of neurons. And so this cell can receive multiple kinds of input from different cells. That input may be synchronous. In other words, the cells may all, the axons may all fire and activate at the same time, or this input may come in asynchronously. And the cell that's receiving the input, this guy here, he needs to do some sort of computation to decide what to do with all that information coming in, whether it comes in all at once or at different times. And the job of the neuron then becomes that of integrating all that information at the different synapses and computing all that input and deciding what kind of output to put out. So it's getting multiple inputs at multiple places on its dendrite, even on its soma. It could be receiving input. Even closer down here towards the axon, it could be receiving input. And it has to integrate all of that and make a computation and decide whether there will be output. So what does the output come in the form of? There's only one form of output that this neuron will give, and that is action potentials. And those will be triggered here in the axon hillock. So ultimately what we're saying is the neuron gets all of this input from different places, and depending on the combination of all that input may or may not fire an action potential. So let's look at some examples of how this could work. So let's say that these two neurons here fire, and they cause the opening of cation channels in the postsynaptic membrane. Then what we'd have is positive ions flowing into the cell that could depolarize it. These positive ions will come into the cell and diffuse throughout the cell, and they may even diffuse all the way down into the axon hillock. And so these sodium ions may diffuse down, and they may depolarize the axon hillock, maybe to threshold. If they do, then we'll get action potentials we'll get an action potential in the axon that will propagate down the axon. But what if at the same time as these two neurons fire, this neuron fires here, and it opens an anion channel? And so when it opens, we have negative charges flowing in. Then in that case, we've got a combination of positive and negative charges coming into the cell and diffusing to the axon hillock. And when they diffuse there, now with those negative charges, there is less of a chance that the membrane potential will reach threshold. So there's less of a chance that this neuron will fire action potentials. Now, will it or won't it fire depends on the relative levels of all of these inputs. If there's more depolarization than hyperpolarization, then we'll probably get closer to threshold. If there's more anions flowing into the cell than there are cations, then we'll probably get further away from threshold. And the cell basically is making a computation based on those different types of inputs. Let's say that this input here, let's say this neuron over here, actually activates a G protein coupled receptor, so a metabotropic receptor. Well, when these guys are activated, they don't let in ions, but they can change the cell. They can actually change other channels and other proteins in the cell. They can make second messengers, and those second messengers can go on to change things that don't directly change the membrane potential, but make the membrane potential more or less likely to depolarize. So, for example, maybe these second messengers close channels, and by closing the channels, they make the membrane potential more likely to depolarize. Or open channels, and they make it more likely to hyperpolarize, or vice versa. And so that's another complex input that can come into the cell that, combined with the other inputs, causes the cell to make a complex computation to decide whether to have an output. Again, the output would be in the form of an action potential. And the action potentials themselves, either happen or they don't. They're all or nothing. But there can be some variation. There can be variation in the frequency that they happen. So you either trigger an action potential or not, and you either trigger high frequencies of action potentials or low frequencies of action potentials, or somewhere in between. And each of those is a different message that the neuron is sending on. If it doesn't send any action potential, then of course it's not sending any message, but if it fires a single action potential, that's one message. If it fires low frequencies of action potentials, one right after another, that's another message. If it fires high frequency action potentials, one right after the other in quick succession, then that could be yet another message that the neuron sends out. But the message itself, the essence of the message, is an action potential or multiple action potentials. But the input, so that the output is action potential, but the input is all of this stuff coming into the dendrite and the soma on this neuron, which can be depolarization, hyperpolarization, or metabolic changes with G protein coupled receptors. All of these can be inputs that can change what the cell does, how it depolarizes or not. Okay, I've drawn a neuron here with a synapse, and this is a chemical synapse, and I want to talk to you about one of the things that can affect the computation that the cell makes. Again, the computation is really made right here in the axon hillock. Do we or don't we have enough depolarization to reach threshold? That's really the question that the axon hillock asks, if it could ask a question. One of the things that can affect that is how much depolarization happens at the dendrites, at the synapse. So the more depolarization that happens here, the more likely it is that those positive ions will diffuse to the axon hillock and depolarize it to threshold. So what determines how much depolarization happens there? Well, what could change how much a postsynaptic membrane depolarizes? Okay, here I'm zoomed in on that synapse here. And I've got my vesicles here in the presynaptic neuron, and they're filled with neurotransmitter. And I've got my receptors here. Let's call this an acetylcholine. Let's pretend that this is a synapse where acetylcholine is going to be released. So let's call these guys ionotropic cation channels. And so they're gated. They're neurotransmitter gated or ligand gated. In this case, we'll call them acetylcholine gated receptors. And so when they open, they will let sodium in, a cation. So here's acetylcholine in the vesicle, and when it's released, when this fires an action potential, and we've got depolarization in the axon terminal here, calcium will come in. It will cause these vesicles to fuse with the membrane, and they'll release their contents into the synaptic cleft. So what can cause more depolarization inside this postsynaptic membrane? Hopefully you answered more channels opening. The more of these the greater number of channels that open, the more sodium can come in. We've talked about this over and over again. The more doors that are open, the more you can flow through. So the more doors we open, the more we can get sodium to come in and depolarize this. So we can increase the amount of depolarization that's happening in the dendrite there. How do you get more doors open? Well, you would get more neurotransmitter released. The more neurotransmitter you have in the synaptic cleft, the more receptors you'll activate and the more doors you'll have open. So how do you release more neurotransmitter? What will cause more vesicles to be exocytosed and release their neurotransmitter contents? Well, going backwards, the more depolarization you have in the axon terminal and the more calcium you have coming in to the cell, into the axon terminal, the more calcium, the more vesicles will fuse with the membrane and release their contents. So hopefully by going backwards here, you see that we can increase the depolarization of the postsynaptic neuron by increasing the depolarization of the presynaptic neuron, its axon terminal, by letting more calcium in. It actually turns out that in a presynaptic neuron, in an axon terminal, the vesicles that are found there all contain about the same amount of neurotransmitter. And so when we measure, when the neurotransmitter is released from the vesicle, each vesicle will release about the same amount of neurotransmitter. And what that means is that the postsynaptic potential in the postsynaptic neuron is quantized in that the amount of depolarization depends on the amount of vesicles released. And one vesicle will release one quantum of neurotransmitter that will cause a certain amount of depolarization. And if two vesicles release their contents, then you'll see double the amount of depolarization. So let's look at the postsynaptic membrane here and look at the membrane potential here in the postsynaptic membrane. And let's pretend that this vesicle fuses with the membrane and releases its contents. It will bind to a certain number of receptors on the postsynaptic membrane and open them, and in doing so will create an EPSP. And that EPSP will rise and then sink again as the neurotransmitter binds and activates the receptors and sodium comes in. And then as the neurotransmitter releases from the receptor, those doors will close and sodium will stop coming in. So this is called an EPSP, an excitatory postsynaptic potential. And what's interesting is that if we depolarize the inside of this presynaptic neuron a little more and release two vesicles, so this one, let's say, releases its neurotransmitter, we will essentially double the number of receptors that get activated, and therefore will essentially double the amount of depolarization that happens. So with two vesicles, if this is with one vesicle, with two vesicles releasing their contents, the EPSP would be exactly double that. So that lower hill was one vesicle, and now this is with two vesicles releasing. And so what this means is that you can actually determine how many vesicles were released with any at any point in time by measuring how big the EPSP was. If you know how much of an EPSP one vesicle causes, then you just measure how big the EPSP is, and if it's 5 times as big as one vesicle, then you know 5 vesicles released their contents. So this is what your book refers to as the EPSP being quantized. It just means that depending on how big that EPSP is, you can actually calculate how many vesicles release their contents, because you know what one vesicle releasing is. These guys are called mini EPSPs. A mini EPSP is when one vesicle releases its contents. The presynaptic neurons will often just spontaneously release a vesicle. And so if you just sit there and measure the postsynaptic membrane potential, every once in a while you'll see a little blip in the membrane potential. And that's because one of these vesicles just accidentally released its neurotransmitter. It probably happens because maybe a little bit of calcium leaks in somehow, but it releases a neurotransmitter and it depolarizes the postsynaptic membrane a certain amount. But that's just one vesicle being released. When this neuron actually activates, it will be much more depolarized, and so the postsynaptic potential here will be much bigger. It'll be some factor bigger than a single mini APSP, like 5 times bigger or 6 times bigger or 200 times bigger. You know the size of a mini EPSP, and so if you measure the size of an EPSP, it'll be some multiple of a mini. The way to depolarize this postsynaptic membrane more then is to release more neurotransmitter from that presynaptic terminal. And the way we release more neurotransmitter from that presynaptic terminal is for that presynaptic terminal to be depolarized more. And there's one, so how would you do that? How could you get this? How could you increase the amount of depolarization in the presynaptic terminal? One way you could do this is through a process called temporal summation. So I'm going to show you a picture from your book that shows temporal summation. So here I have an axon synapsing onto this dendrite, and what this neuron did was fire 3 action potentials right in a row, one right after the other. So a high frequency train of action potentials, and all 3 of these are traveling down the axon, and when they get to the axon terminal, the first action potential that comes into it will cause it to depolarize. And what do we have happening in the axon terminal? Remember, we have the action potential travels down and it spreads out in the axon terminal, and it opens up voltage-gated calcium channels. And calcium can flood into the axon terminal. But what is happening at the same time as calcium is coming in? Is there something removing calcium too? Yeah, remember there's a calcium ATPase also here that is pumping calcium out at the same time, because ultimately we want to return the calcium levels to low levels inside the terminal. So you've got these 2 things happening at the same time. You've got calcium coming in through the voltage-gated calcium channels, but you also have calcium leaving through the pump. Now, if that action potential had come down all by itself, it would have caused depolarization, calcium would have come in, some of the vesicles would have exocytosed their contents, neurotransmitter would go across to the postsynaptic membrane, and then it would stop. But when we have a second and a third action potential coming in right behind that first one, the calcium in the axon terminal will get higher and then even higher than it would with just one action potential. Because what's going to happen is with that second action potential coming in, it will come in and cause even more depolarization in the axon terminal, which will open up even more calcium channels that weren't open before, and calcium can come in and grow to even higher levels before the pump has a chance to pump it back out. And so this is called temporal summation because we're basically building on the action potential that came in before. And so if you have a lot of action potentials in a row, the inside of that axon terminal can become very depolarized and you can have a lot of calcium in there and thereby release more vesicles than normal than you would with just one action potential. So if you measure the postsynaptic membrane or the synapse there, you'll see that with that first action potential you got some depolarization, but before it was able to go all the way back down, that second action potential released more neurotransmitter, which caused a higher depolarization that built on the first one, and then the third action, and before that made it back down, you had a third action potential that brought the membrane potential even higher. So with each of these, you're getting, what's happening is that in the postsynaptic membrane here, in the postsynaptic cell, you're getting depolarization coming in to the cell, but before that depolarization has had time to diffuse away and dissipate, more depolarization comes in the form of sodium, and before that has had time to dissipate, even more comes in because of that high frequency of action potentials that was fired in the presynaptic neuron. We just learned then that one way that you can increase the amount of depolarization in a dendrite is by releasing more neurotransmitter all at once. And that could be by firing action potentials one right after the other in an axon. The other thing that you could do, or the other thing that you could see happen to cause more depolarization, is for there to be a second neuron that synapses at the same place. So it synapses at a very close by place on the dendrite. They can't be in exactly the same place, but they can be very close. And these two axons, axon 1 and axon 2, can both fire at the same time and release neurotransmitter at the same time. And so then you'd have a lot of neurotransmitter in the synaptic cleft, and it could activate more receptors because you have two neurons firing at the same time, and that could open up even more doors than just with one neuron firing. And that is called spatial summation. So spatial summation is when you have multiple neurons all synapsing in the same general region of the postsynaptic cell, and they all fire at the same time, and so their action potentials all reach the dendrite at the same time, and they all release neurotransmitter at the same time, and they all activate receptors in that that small area at the same time, causing a large amount of depolarization. And what will that do? Well, either with temporal summation or spatial summation, what you've done basically is increase the depolarization in that dendrite and biased the computation. The neuron's making a computation, right? You've basically biased it towards depolarization because you've basically put a lot of positive charge into the cell. Whether or not that causes the axon hillock to depolarize to threshold is yet to be seen, but certainly that summation is going to help the chances of it getting to threshold. All right, now let's talk about something called the cable properties of dendrites. So let's say that we have just allowed the dendrite here has just become depolarized because this neuron released a neurotransmitter that opened channels that were cation channels, and this dendrite depolarizes. Now, how likely is it for that positive charge to be able to diffuse all the way to the axon hillock where it can bring the neuron to threshold and cause it to fire? Well, not only we just learned that the amount of depolarization, whether it's more or less, will change your chances of depolarizing to threshold at the axon hillock, but also the cable properties of the dendrite, in other words, the structural shape and contents of the dendrite can change how far that positive charge gets. So let's look at a dendrite and think of it as a hose. So I'm going to zoom in on this dendrite here, and we're going to think of it as a long tunnel. And so here's the synapse right here, let's pretend. And so depolarization has come down here, and the axon hillock is down here. What would improve the chances of that positive charge being able to get down to the axon hillock? Well, there's a couple things. Actually, there's three things that we're going to learn about. One thing is called membrane resistance. So let's think of this membrane as a hose, and the positive charges as water, and we want them to get down to the end of the hose. What if that hose has a bunch of holes in it? Your water's not going to get down. It's not going to get very far. Membrane resistance is basically how leaky the membrane is. So if this is a dendrite and this is the dendrite's membrane, how many holes or channels are here that would allow the positive charge to leak out? If there are a lot of these, if it's very leaky, then the positive charge isn't going to get as far down. It's going to leak out. So instead of getting down the dendrite, it's going to leak out instead. So membrane resistance is how leaky the membrane is. High resistance means low leakiness. So if my membrane has a lot of resistance, that means it's not very leaky. And so if the membrane has a lot of resistance, it means that a positive charge can travel farther. Okay, the second factor that determines how far it can travel is called internal resistance. All right, let's say that your hose is full of dirt and mud. Will that help water flow down? No, the dirt and mud will block it. So dendrites don't have dirt and mud in them. Did I say blood or mud? Anyway, dendrites don't have dirt or blood or mud in them. They have proteins. But those proteins can get in the way of positive charges flowing down because proteins have negative charge to them. And so if you have proteins in here that are in your dendrite, okay, and it's negatively charged, this positive charge diffusing down the dendrite won't make it as far because it will be neutralized by those negative charges on the proteins. And so this is called internal resistance when you have a lot of proteins in the dendrite. And they will prevent the charge from getting down as far towards the axon hillock. So more resistance means less distance that the ion can travel. So we saw membrane resistance. Actually, if you have more membrane resistance, that actually helps you get down further. But internal resistance, that actually prevents you from getting down further. So to get way down the dendrite, what you want is high membrane resistance and low internal resistance. And having those two things will increase what we call your length constant. The length constant is just how far the positive charge can travel. And so the greater the length constant, the further that charge can travel. So membrane resistance will help it travel farther, so it will increase the length constant. And bringing down your internal resistance will also increase the length constant. It will allow that positive charge to get further down the dendrite. The last thing is the diameter. So how wide is your hose? How big of an opening does your hose have? The wider the opening, the better it is for water to travel down. Same thing for these electrical charges. The bigger the hose, the bigger the diameter, the better they can travel down. So diameter and increased diameter will also increase your length constant and allow that positive charge to travel further down the dendrite. There's one more thing that can help the positive charge flow down further and increase the length constant. And that is, what if in the dendrites you had voltage-gated sodium channels? We're used to seeing these in an axon, but they actually can appear in dendrites too. So voltage-gated sodium channel. What will these do? Well, when positive charge comes in through these ligand-gated channels here, it will diffuse in and it could bring this channel to threshold. If it does, it would activate and it would let in even more positive charge. And so that would give a boost to your signal. And so now you'd have a better chance of that positive charge getting to the axon hillock. So when a dendrite has voltage-gated sodium channels, we call that an excitable dendrite. And of course, that would cause the positive charge it would make it more likely for that positive charge to get to the axon hillock. It turns out, in fact, that if you measured the membrane potential in an excitable dendrite, you'd see something like that. So in the dendrite, you could actually see an action potential in an excitable dendrite. you could actually see an action potential travel down the dendrite. And an action potential would really, if it's traveling down the dendrite, it's really going to increase the chances that you'll get an action potential in the axon hillock, because that's a big jump in positive charge that's happening in the dendrite. And so it makes it very likely, much more likely, for that positive charge to make it all the way to the axon hillock. All right, let's talk about a way that you can that cells can actually prevent a depolarization from getting to the axon hillock. Our brain does really complicated things. We're very complex creatures. And in some cases, we may get input to a neuron that activates it or depolarizes the neuron, but we also might get some input that's trying to inactivate the neuron. So the neuron may be getting conflicting input. So I'll just draw a real simple neuron here. This axon here may be giving input that depolarizes the cell. This one over here may be giving input that hyperpolarizes it. In some cases, these two neurons can be found on the same dendrite. And in that case, they're going to neutralize each other. And this is called shunting inhibition. So here we have a dendrite. Here we have a long dendrite on a neuron. And here's one synapse that's excitatory, so it'll depolarize the dendrite. And further down towards the soma, we have another synapse that is inhibitory, so it will open anion channels. This one up here will open cation channels. This one will open anion channels. So this one will hyperpolarize the membrane, and this one will depolarize the membrane. So what happens if just the excitatory one fires? Well, that positive charge will come in, and if I'm recording down here in the soma, the postsynaptic potential, it looks like here I'm recording two places. I'm recording right at the synapse and then right down here in the soma. So if I record right at the synapse, I'll see a big postsynaptic potential, excitatory. If I record down here at the soma, I'll also see an EPSP. It won't be nearly as big, though, because the ions, when they come into the cell, they diffuse in all directions. And so here I'm measuring right at the source of the ions where they're coming in. So I'll see a big EPSP here. Down here, it won't be nearly as big because some of those ions have diffused to other places. But it can even be reduced. That depolarization that I see in the cell body can be reduced even more if I fire that inhibitory, if that inhibitory synapse becomes active at about the same time. Then what happens is those positive charges flow into the dendrite. But what happens right here is now I've got negative charges, anions, flowing into the dendrite too. And so they'll neutralize each other. And so when I measure in the cell soma, I don't see any of that blip. It's gone because those positive charges didn't make it in there at all. This is called shunting inhibition. And this may be, where would this be used, or how would this be used? Well, perhaps this is used in situations where we become desensitized to certain stimuli. So think about a perfume or a cologne that you put on. At first you smell it, but after a while you get used to it. Even though the cologne is still there, you've become desensitized to it. So what might have happened is when you first put it on, your nose sent you information that activated this neuron, and you were like, oh, I smell something. But then after a while, some other neuron became active and said, we're done smelling that, so we need to shut that down. And so even though the cologne is still exciting and saying, hey, there's still a smell there, this other neuron, which is an inhibitory neuron, is coming in and saying, yeah, I understand that, but we need to, we can't constantly be smelling the cologne. We've got other things that we need to think about as neurons. And so this one will become active, and so this neuron here that's responsible for telling you, giving you the conscious experience of smell, is turned off and you don't smell anything anymore. That might be one reason that the brain would use shunting inhibition. Okay, the last topic is called in your book modulation. Let's look at this dendrite again. And we talked about its membrane resistance. We said if a membrane is very leaky, if it has low resistance, then the positive charge won't be able to travel as far. So we have these channels that may allow leak to happen, and the positive charge can leak out. That was supposed to leak out, not leak in. What if we could close those channels? What if there was a way to actually close them? Then instead of that positive charge leaking out, you could keep it inside the dendrite, and it could get further down the dendrite towards the hillock. There are ways you can close those channels in certain circumstances. And often the way those channels are closed is with G protein coupled receptors. So there are some G protein coupled receptors. And I'm drawing it like that because those G protein coupled receptors are a long polypeptide that weaves in and out of the membrane kind of like a snake. And when that G protein coupled receptor is activated by its ligand, and this is the G protein coupled receptor, it activates second messengers. It activates enzymes that turn on second messengers. So let's remind ourselves how these work. These receptors are activated, they change shape, and what they do is they cause a G protein to become active. This G protein that can then activate itself another enzyme. And this enzyme can make second messengers. And these second messengers can close these can close these channels. So how could they do that? I'm going to have to introduce to you now something called a kinase. So a second messenger is a small molecule, and it can activate. It carries a message. That's why it's called a second messenger. It can activate other proteins that do things. And one of the proteins that it can activate is called a kinase. And a kinase's job is to use ATP to add a phosphate to other proteins. So a phosphate, if you think about ATP is adenosine triphosphate. So it's this molecule that has 3 phosphorus atoms on it. And these phosphorus atoms have oxygens around them. And these oxygens, I think that has another one. And these oxygens, I'm not drawing it perfectly because I don't know the structure, but it's got adenosine and then 3 phosphates. That's ATP. And these phosphates are negatively charged. The oxygens are on the phosphates. And this thing right here is called a phosphate. And this would be another phosphate, and this would be another phosphate. So it's adenosine triphosphate because it has 3 of them. What a kinase does is it pulls off one of these phosphates and adds it to something else, like a channel. It can actually covalently attach that phosphate. It can remove it from ATP and attach it to a protein. It will attach it to an amino acid in the protein. So the amino acid has a place on it where a phosphate can be attached. And so the kinase takes it and sticks it on there. Because it's negatively charged, it's like attaching a big negative charge to the protein. That's going to push on positive charges that are in the protein and change the shape of the protein. So let's pretend this is a channel, let's pretend this is a potassium leak channel. That phosphate, that negatively charged phosphate that the kinase put on there, can actually change the shape of the channel because in the walls of the channel there are positively charged amino acids. And so that phosphate can push on them and cause that channel to change shape and even cause it to close. Okay, so if that kinase acts on this guy, and it adds a phosphate there and causes this to close, and acts over here and causes this to close, that kinase could close all of those potassium channels. So what it does again is it removes the phosphate from ATP and attaches those to other proteins like channels. And when that protein gets a phosphate attached to it, we say that that protein is phosphorylated. And a phosphorylated protein will change shape. And if it's a channel, it may close the channel. When ATP is used, by the way, it's the remaining part has two phosphates, so we call that adenosine diphosphate, ADP. But that's kind of the byproduct of what when a kinase acts. So a kinase will use an ATP, add a phosphate to a channel, and then it'll get another ATP and add the terminal phosphate to another channel, and then it'll get a third ATP and add a phosphate to that other channel. And that's how it phosphorylates things. And it will do that as long as the second messenger is around, because the second messenger is what was turning that kinase on. So as soon as the second messenger is gone, the kinase will stop. But the kinase has already done the dirty work, and now I have 3 phosphorylated channels. And that phosphate can stay on there for quite a long period of time. What did we just do to the membrane resistance of that dendrite? We just raised it. The membrane resistance just went up, because now, instead of that positive charge leaking out, potassium is positively charged, right? Instead of positively charged potassium leaking out, it can get further down the axon. And if sodium's coming down, the dendrite, if sodium's coming down the dendrite, this positive charge here with potassium that stays in will neutralize any negative charges that are here and allow sodium to get by without getting neutralized. And so that sodium can get down farther because we've kept potassium in. We keep potassium in, it keeps the inside of the cell more depolarized, and so that increases the chances the axon hillock will become more depolarized. The axon hillock doesn't care whether we've got potassiums or sodiums. It just looks at positive charge, and both of those have a positive charge. So whether we've got potassium coming down the dendrite or sodium, we're increasing the positive charge. And that kinase can affect it by phosphorylating those channels and closing them. Okay, there's actually a specific enzyme and specific second messenger that do this. The specific enzyme is adenylyl cyclase. The specific second messenger is cyclic AMP. And the specific kinase that does this is called protein kinase A. Just because I added the names doesn't make it any more complicated, except now you know their names. But the concept is then, this ligand, this neurotransmitter, it's not going to open doors, but it's going to change the inside of the cell. It activates a GPCR, which activates a G protein, which activates adenylyl cyclase, which produces cyclic AMP, which activates this kinase, who then closes all of these doors and makes the inside of the cell more depolarized. That lets the depolarization get further down the track. Adenylyl cyclase, by the way, uses ATP to produce cyclic AMP. So it uses ATP too. Lots of things use ATP. So we could say about this GPCR and its activation that it made the dendrite more excitable because it increased the length constant of the dendrite.